So Jessica's presentation was a phenomenal primer to what I'm going to show you today. So I'm going to point to two messages or a little bit add to the discussion we were just having with Josh, Joe, and Jessica. That really to annotate metabolomics data, you need your own knowledge of your system to kind of, if you have those top 10 candidates, which one could be the candidate, that's the actual candidate, that's where your biology and biochemistry knowledge comes in. That can my system make this molecule? Does my system have the genes to make this molecule, right? So I'll add to what you said. If you're a biologist, God bless you. I think the biologists need to be doing metabolomics. And that's where my training comes from. So in my PhD, I was a biochemistry major. My extent of use of mass spectrometry was I would incubate my peptide with my enzyme, look for product formation. Or my peptide was post-translationally modified. I would use tandem MS to figure out where the cycles are in my peptide. So that was the extent of use of mass spectrometry for me during my PhD. In my postdoc is when I learned metabolomics. I was introduced to DDA, the term DDA, for the first time during my postdoc. So as a biochemist who's moved into metabolomics, I really appreciate the power of combining microbiology, biochemistry, and metabolomics and enabling new discovery. So today I want to give you a little bit of a flavor of how metabolomics has now advanced to a point where we can link mechanistic biochemistry with chemistry and phenotype, enabling discovery of new molecules, new functions, new pathways, and is really shaping our knowledge of cell biology, physiology, medicine, ecology, you name a field, and we are shaping the understanding of that field. So I'll highlight in my talk today application of a number of things that Jessica mentioned. So I'll highlight a number of tools. I'll not go into the details of the tools. I'll highlight when would you use this tool, how would the output of this tool combined with your biochemical knowledge enables you to annotate stuff. And then tomorrow, Jessica Deutsch, my fifth year graduate student, will actually walk you through how to use those tools. So she's actually going to go to the servers, teach you where to click buttons, and she'll have a cookbook recipe for you that you can later practice the files she shares with you and try to annotate molecules. So the application I'm going to show you today is using microbial metabolomics. My lab uses metabolomics to study microbe. How do they cause infection? How do they become antibiotic resistant? How are they persistent? How they interact with host? How do they interact with other bacteria, and so on and so forth. So I'm not going to go into too much detail of the biology. I'll only go into the biology when I need to explain why I annotated this molecule as X. Okay? So we study these bacteria called Burkholderia cepacia complex. So why did we want to apply metabolomics to study this bacteria? When staph causes infection, it's always aureus. When Pseudomonas causes infection, it's always aeruginosa. But when Burkholderia causes infection, 20 different species of Burkholderia can cause infection. And depending upon which species is causing infection, the treatment outcome is very different. So I, as a biochemist turned metabolomics person, wanted to explore, can I use metabolomics to kind of identify signatures of metabolites that are linked to treatment outcomes or linked to species that cause more fatal disease. To do that, we study bacteria in a model system, and towards the end of my talk, I'll highlight why it is important to do metabolomics first in a model system, then in a actual human sample. So the patients, the lungs, from where our bacteria come from, are filled with mucus. So we synthesize this mucus in the lab. It contains the protein mucin. So if you were doing proteomics, you would see a lot of mucin in there. It contains lipids that are surfactants, DNA, bacteria, humans, neutrophils spit out a lot of DNA, creating this gooey mucus, and then average nutrient content. So this is our model system. But if you take a human sputum from these patients, the most abundant molecule in there is antibiotics that these patients are taking at the time of treatment. And if you take 1 ml of patient sputum, there's only 1 microliter bacteria. So the signal from microbial molecules is really, really small. That's why we want to first study them in a synthetic sputum, inventory the molecules that are important, that are bacterially derived for infection, and then go do more targeted methods to identify them in patient samples, for example. So we culture our bacteria, we expose them to antibiotics, we do untargeted metabolomics. We are moving into transcriptomics. We are, with our collaborators, learning how to do transcriptomics so we can correlate this to to kind of make sense of disease prognosis. Jessica gave a wonderful primer, like I said, for my talk. So I don't have to go into too much detail of workflow, but I want to remind you just a little bit, what do you do once you have acquired the data? And remember, Jessica mentioned that there's a lot of bias in how we acquire data, right? The column you use, the solvent you use for extraction, the instrument you use, right? So we are past that. We've acquired data. We're happy with what we have. And then Jessica mentioned you detect features, right? And you can calculate area under the curve for each feature that is detected, and then you have this Excel file that she showed. Then you apply multivariate or univariate statistic analysis, the same kind you apply for proteomics, volcano plots, random forest, PCA, PLSTA, to figure out how your sample sets are grouped. For example, we are interested in how the metabolic profiles of different Burkholderia species are. And then if they are different, what contributes to the differences. So you can rank metabolites that account for this variability, right? There are several methods to do that: random forest, PLS, support vector machines, several other methods, modeling methods. And once you have this ranked metabolites, that's when the challenge starts, because now you have to identify what those metabolites are, and that identification allows you to make biological interpretations. So we have to overcome this bottleneck. And highlight of my talk and the tools that Jessica is going to show tomorrow are going to be focused on how do we identify, and then how do biologists and biochemists help us in validating those identifications. So as Jessica mentioned, we use tandem mass spectra. Now by third day of summer school, I hope everybody knows what a tandem mass spectra is. And then, as Jessica mentioned, you look for them in spectral libraries. If you get a match, you can name a molecule. And we use today I'm going to leverage additional techniques that allow us to, or bioinformatic methods, that allow us to propagate these annotations. Or if you don't have any annotations, how class predictions, substructure discovery allows you to then decode this dark metabolome. So one of the methods I'll start with is called molecular networking. It's based on the premise that if there is spectral similarity, that implies structural similarity. So if there are two molecules that are structurally similar, some of the fragments are going to be identical in the masses to charge ratio, right? So for example, these green fragments are identical to these blue fragments, right? But the other fragments, these two here, are shifted by the mass of functional group that's different between the two molecules. So hydroxyl is 15.99. So that's where you're seeing the shift in these two peaks. Okay? So if you know this molecule, you can annotate this one. Okay? So computer scientists are very good at quantifying similarity. Nuno Bandera at UC San Diego, Peter Dorestein came together. They wrote an algorithm which allows you to quantify this spectral similarity in form of a cosine score. But as a biochemist, I like to see this. I don't know what the code is, but I can represent this spectral similarity in the form of these networks, where each node is a spectra, and two connected nodes are spectra that are similar. So they're similar structures. And if there's a node hanging out here by itself, the spectra of this molecule is not similar to any other molecule in your data set, okay? So if two things are similar, they'll be connected. If they're not connected, they're unique, okay? And then in addition to molecular networking, I'll highlight, so if you don't know what this molecule is, then what do you do? Then we use substructure prediction, chemical classification to decode the unknown metabolome. Okay. So we cultured, like I said, Burkholderia, different species cause infection. Depending upon what species you have, your infection outcomes are very different. So Cynocepacia causes the most fatal infections. So we cultured 4 different species. For each species, we cultured 3 different strains, and we looked at their metabolomes. And we pulled out features that were first unique to B. Cynocepacia, the most fatal infection-causing species. So shown here is an example. So this is first set of cluster which was unique to B. Cynocepacia. That's another advantage that Jessica will highlight tomorrow of molecular networking, that you can color code, shape code your metadata. So I colored the node based upon in what samples they were detected. So based upon the cyan color, I know that they were only detected in B. Cynocepacia. So then what do you do? Once you have this cluster of nodes, you know they are structurally similar. They are only produced by the bug you are interested in. You look for their fingerprint, their MSMS spectra in databases. So we looked in every single database that Jessica highlighted on her slide and many more that were not on the slide. And we came back empty-handed, no annotations at all. So what do you do now? You can either quit, go work for Thermo and sell Orbitraps, or you can go to the dark world, work in the field of proteomics, where maybe you can annotate 40% of your data, or you can hang around and apply every single tool that's out there that the community has created in the last 5 to 10 years. So substructure prediction, chemical classification to kind of make sense of your data. So that's what we did. We did not quit. I am not working for Thermo. I'm hopefully making tenure next year. So we used this tool called MS2LDA, which was developed in 2016 by Justin Vanderhooft and Simon Rogers at University of Glasgow in UK. So what this tool does is it won't tell you what your molecule is, but it can predict a substructure. For example, if your molecule is this, it can predict that it contains a pentose sugar, for example. So this cluster is a cluster by itself. This cluster is a cluster by itself, no annotations, but they may share a common substructure. So if you can annotate this red node, then you can propagate those annotations to this unknown cluster via the known substructure, okay? So when we applied this tool, and Jessica will show you tomorrow how to use this tool, the substructure came out to be trimethoprim. So trimethoprim is the antibiotic that's first line of defense when you get a chronic Burkholderia cynosephagia. So our patients were on the antibiotic trimethoprim, so we had added it to culture. But Jessica Prenny mentioned to remove the artifacts, you remove your blank. So our media contained trimethoprim, so we had deleted the media background, so that deleted the data for trimethoprim. That's why trimethoprim was not connected here, otherwise trimethoprim would be in this network. So using this approach, we figured out that all of these nodes contain trimethoprim as substructure. That means the most fatal bacteria, cynosephagia, is able to modify the antibiotic. So we thought we are onto a new mechanism of antibiotic resistance, which is not known. And that's unique to this specific bacteria. So we were excited, and we wanted to figure out what these molecules are. And I mentioned for every bacteria, we cultured 3 strains. So we asked, do all of them modify the antibiotic? Turns out 2 of them do, and 1 of them does not. We got lucky that the ones that modified the antibiotic were brown in color, and then the one that doesn't was not brown in color. Now, this is where you need a biochemist or a biologist, right? So I knew how to look at biochemical pathways, so we went to the biochemical pathway of how pigmentation is produced. So it starts with tyrosine. Typically, tyrosine is converted to fumarate and acetoacetate. That goes into the TCA cycle. In bugs that are pigmented, there's a mutation in this gene, so the pathway flux is altered. It doesn't go this route, but goes this route. So you accumulate homogentic acid, oxidize it, and convert it to the pigment. So I thought it's too much of a coincidence that you have pigmented bacteria only producing these cluster of molecules. So pigment may have something to do with antibiotic metabolism. So we brought in our biology collaborator. She made genetic knockouts. So she converted the pigmented bacteria to non-pigmented by swapping out the HMGA gene. And then she converted the non-pigmented to pigmented. We grew them, we did our metabolomics, and we found that only pigmented bacteria metabolized the antibiotic, non-pigmented did not. So clearly pigment has something to do with antibiotic metabolism. And the masses to charge ratio of trimethoprim is about 291. So you're adding something to it. And only pigmented bacteria can add. So the metabolomics side of me thought, maybe there's a unique precursor in pigmented bacteria that can react with trimethoprim, and it's absent in non-pigmented bacteria. So what if I compare the metabolomes of pigmented and non-pigmented, figure out what's unique in pigmented, and see if that's what's added to trimethoprim? So we took that approach. We looked at the metabolomes of pigmented. So same data, you just visualize it in different way. So now we visualize pigmented versus non-pigmented. This is an upset plot. Think of it as Venn diagram. It's just easier on eyes. These are the intersections. So we found that 12% of the metabolome, about 500 molecules, were unique to pigmented bacteria. So again, my student with his undergrad sat down, searched every single database, spent a few days, got really frustrated. No annotations at all. So what do we do now? So then we again called the in silico methods. First, we wanted to focus on 20 to 50 molecules, not all 500. So how do we pick out which ones to go after first? we used a method which is not very well liked, ranking metabolites, random forest or PLSTA. So we put them on a plot, and we first assigned chemical formulas. So Jessica's going to show you tomorrow Canopus. It's the best tool for predicting chemical formulas. It uses both MS1 and MS2 spectra to predict chemical formulas. So from the MS2 spectra, you make fingerprint, and you use your MS1 isotopic pattern, combine it with the fragmentation tree to predict a chemical formula. So you can rule out a lot of chemical formulas because they don't fit your MS2 spectra. So it improves the prediction of chemical formulas. So a common theme was multiple sulfurs, 3, 4, 1. So we thought it's one biochemical pathway likely producing all of these unique set of molecules in pigmented bacteria because they all contain sulfur. Then we used this new tool. Jessica will highlight this tool tomorrow, and I'll also show you a few additional applications of this tool. This tool takes fragmentation data and predicts chemical classes. And Jessica highlighted in her talk today that these tools are really useful, and they are enabling us to decode the dark metabolome. And I'm going to highlight in a bit how they do that. So if you look at the chemical classes, and if you start reading, you read a lot of benzamides, thiazoles, arylamides. So a common theme is that all of these molecules contain sulfur, and seems like they're all aromatic. So then we asked, can we predict a substructure? We were able to predict substructure using the MS2LDA tool that I highlighted. And again, Jessica's going to show you all of these tools, how to use them tomorrow. So we did predict a substructure. We couldn't annotate it. But if you look at the substructure, again, it has multiple sulfurs. And if you look at the carbon to hydrogen ratio, for 10 carbons, there are 12 hydrogens. So that's pretty unsaturated, suggests an aromatic ring, likely, right? So again, now we know that our compounds contain multiple sulfurs. They're likely aromatic. And they're only produced by bacteria that's pigmented. So that's where the biochemist comes in, right? So it's too much of a coincidence that all of these are aromatic too. So could these be the precursors of them? Now again is where you need a biochemist. Well, mass spectrometrists know this too. You can use isotope labels. Since this is a model system where we culture bacteria, we can change the food we give to bacteria, like we can change the media conditions. So we cultured them in labeled isotopes. So like if they come from tyrosine, we used a labeled tyrosine where each of this carbon is carbon 13. Josh talked about isotopes yesterday, carbon 12, carbon 13. So instead of carbon 12, we have carbon 13. And the sulfurs biologically come from typically either methionine or cysteine. So we added S34 instead of S32, methionine and S34 cysteine in our media. And then we acquired metabolomics data, and we called this method as metabolite annotation assisted by substructure discovery and stable isotope labeling. Today, if you are in metabolomics world, and if you come up with a method, if you don't name it, you haven't done your job. So we kind of came up with the name MassILAC. So using this method, we were able to annotate our substructure as being derived from homogentic acid. So this is a decarboxylated homogentic acid which has undergone thiomethylations. So using this knowledge, we were able to annotate the pathway for trimethoprim metabolism. So we start with tyrosine, which is converted to homogentic acid. And we know homogentic acid accumulates in pigmented bacteria because the next enzyme has a mutation, doesn't work, right? So then methionine is converted to methane thiol. Oh, this slide is not updated, I apologize. But methionine is converted to methane thiol, which is basically CH3ASH. So if you're the graduate student working in the lab, you don't need to do GCMS. It's so pungent that your bacteria is making methane thiol. So he did GCMS, he confirmed that it makes methane thiol enzymatically, and that methane thiol gets added to homogentic acid. So you see these 3 thiomethylations. So this is a new metabolite. It wasn't present in any of the databases that we searched, not even structure databases. So completely new endogenous metabolite discovered using simple untargeted metabolomics. And then this is the molecule that gets added to trimethoprim, resulting in its inactivation, right? So using our knowledge of metabolomics, untargeted metabolomics, biochemistry, our ability to make gene knockouts, we were able to figure out a new pathway. So for metabolomics to move forward, you really do need biochemists and biologists to be working with you. So you may have heard of antibiotic resistance mechanisms, very quickly going through them. One of them is antibiotic inactivation. You may have heard of beta-lactamases, opens up the lactam ring, inactivate the antibiotic. Here, acetylation of chloramphenicol inactivates chloramphenicol. So we've discovered a new pathway using our untargeted approach, where addition of this thiomethylated homogentic acid, which is specific to pigmented bacteria, to trimethoprim results in its inactivation. So these bugs that were isolated from lungs of patients who were on trimethoprim had this unique mechanism of antibiotic resistance. And this was limited by methionine. So if in our culture we add more methionine, we make more methane thiol, we make more of this molecule, and we inactivate more of trimethoprim. And in human sputum, it's very rich in amino acids. So the MIC values that you would get in vitro versus in vivo would be very different. Because in vitro, you're limited by methionine in your media, but in a human, you're not. Right? So we show that untargeted metabolomics allows us to link biochemistry, phenotype with chemistry, with clinical implications. So now, this was the plot I showed you where we had multiple sulfur-containing compounds that were unique to pigmented bacteria. Some of them are metabolism of trimethoprim, but using the same mass SILAC approach, we annotated several of them. The top ones are known antibacterial, antifungal compounds, which were reported in 1960s in some chemistry papers, and we identified also a few of the known ones. So let me summarize what I showed you so far. I showed you that untargeted metabolomics allows discovery. I showed you the discovery of a new metabolite, methylthiolated homogentic acid, and I also showed you how, combined with biochemistry and molecular biology, that allowed us to link phenotype with function. So pigment production resulted in unique metabolic profiles that were responsible for modification of trimethoprim, providing these bacteria pathogens with enhanced functions. Okay, so what can we do now? We still have a lot of annotated spectra. How do we go about them? So what else can you do with your data? Well, you got to ask a different question. So the question we asked was, so we know antibiotics are modified by this bacteria. And researchers have been talking for decades how antibiotics act as signaling molecules. We know there's been proteomics work, transcriptomics work, which shows that when you add antibiotic, bacteria changes its gene expression, right? So how does that lead to changes in metabolic profiles, and how does that relate to bacteria interacting with other bacteria in the lungs or with their host? So then we took, again, the same exact data set we had, but we visualized it in a different way. So we performed a simple hierarchical clustering analysis. Here is just the name of the strain with trimethoprim without. And we identified, just looked for visually clusters that were unique to either presence of antibiotic or absence of antibiotic. So these are unique responses to antibiotic, right? So, and they are strain dependent. So K56.2 shows this, J2315 does not, CF54, so on, does not, right? So this tells us they are likely the specialized metabolome of a pathogen. So for example, tryptophan, everybody has it. But there are some molecules like vancomycin, which only certain bacteria can make, others can't. So that's called the specialized metabolome, which has important function in bacterial physiology. So this suggests that these are natural product vancomycin-like molecules that are produced by strains in presence of antibiotics. So we wanted to annotate them. Again, we start the process of searching databases, come back empty-handed, get frustrated for a day, and then start with in silico tools. But when we first published this paper in 2020, I was 3 years into my tenure. Like Joe said, sometimes career makes us make decisions. So we just published them as unknown molecules. I needed papers. We published them in ACS infectious disease as unknown molecules. But then in 2020, the tool came out, the tool Canopus, which I mentioned predicts chemical classes from fragmentation spectra. So Canopus, and Jessica's going to show you the exact same example tomorrow, clicking buttons on the server and show you how to predict chemical class. So Canopus predicted the chemical class for all 4 compounds as pyrazine ring-containing compounds. So we don't know what the compound is, but we know that they contain pyrazine, or they might contain pyrazine, right? Chemical classification can be wrong as well. But Jessica showed that 90% of the time, chemical classification is correct. Annotation could be wrong, but chemical classification does a good job, or chemical class prediction does a good job. So then we did a very targeted search in the literature or databases for pyrazine-containing compounds. And we focused our search to microbial molecules, because a bacteria is making this molecule in our case. So we came across these compounds called pyrazine N oxides. How these molecules are made biosynthetically is known. So we looked into our genome and we found the pathway for these molecules. So that's an added layer of annotation. So our bacteria has the genes to make this molecule. And then we got lucky. Bo Lee's lab at UNC Chapel Hill had just isolated these molecules and published the structure of these molecules. So I'm glad we didn't isolate them because we both would be isolating the same molecule and putting the structures out there at the same time. And if you're an organic chemist and you get a structure of a known molecule, they're like big whoop. Right? So I'm glad we didn't spend 6 months to a year isolating this molecule. We relied on the in silico methods. Bo was very kind. She sent us an authentic standard of this molecule because she had isolated them, done NMR for structural characterization. So we did just spectral matching of the MS2. We also spent $25K putting UV-DAD detector on our mass spectrometer. Now their scan rates are fast enough. They can keep up with MS2 acquisition or MS1 acquisition rate. So the UV visible spectral match, MS1 match, MS2 match, Jessica mentioned is a level 2 annotation. So we confirmed this using level 2 annotation that that's the compound we have. Interestingly, in plant pathogens, if you knock out the genes for this pathway, they don't cause infection in plants anymore. So Pseudomonas have the same genes. And if you knock out these genes, they do not cause infection in plants. And in humans, these molecules are only produced when you add antibiotics. So we are adding antibiotics to treat this bacteria, but they are making them more virulent. And metabolomics allows us to make that connection. I'm not going to beat the horse, but there were many such molecules we describe in our paper which were either produced in presence of antibiotics or absence. But here I want to highlight one which we haven't isolated so far, but it's a halogenated molecule. So Josh mentioned yesterday, we have C12, C13, they have different abundances. Similarly, halogens, chlorine has a 35 or a 37 isotope. They have different abundance, so 75% versus 25%. So if you have one chlorine, these are 2 Dalton in mass difference, so 486 versus 488. The peak ratio, height ratio is 1:3, 75 is to 25, right? So 1:3 ratio of 486 to 488. And then there's a mass defect, 9689 versus 9659. So with high resolution mass spectrometers, using this profiling of the isotopic pattern, we know for sure that this has a halogen in it. So a very commonly known halogen-containing compound is vancomycin, or antibiotic of last resort. I mentioned that to highlight that halogen-containing compounds have important functions in bacteria, right? They're important they have important pharma structures. So this may be an important molecule in the physiology of this bacteria, and again, it's only produced in the presence of antibiotic, okay? So then the biochemist in me wanted to figure out why. Why are antibiotics inducing the production of these compounds and making the bacteria more virulent, or how? So again, we went into our mass spec data. We figured out some of the molecules that were different between antibiotic presence versus absence. And some of these molecules were these signals here. These are called acyl homoserine lactones. So bacteria have a phenomena called quorum sensing, where you have a gene for a synthase that makes this molecule, which when bound to receptor, regulates gene expression, okay? So we thought maybe this is the way it's responding to antibiotics. So we made knockouts in every single of this gene to figure out whether that's how bacteria is responding to antibiotics. Importantly, B. cenocepacia, the bacteria that we are interested in that causes most fatal infections, only this bacteria had an orphan receptor. So there's no gene for synthase. It doesn't make acyl homoserine lactones, but it has a receptor that can talk to these two pathways. So we made genetic knockouts, acquired untargeted metabolomics data, and found something really cool. So we found that wild type in absence of antibiotic doesn't make the molecule. In presence of antibiotic makes it. If you knock out this orphan receptor, it makes the molecule no matter what. So that means this receptor is a repressor of gene expression. When you add antibiotics, we don't know how, antibiotics interact with this transcriptional repressor, removes the repression, and you can see the production of this molecule. So using, again, untargeted metabolomics and biochemistry, we've not only discovered new molecules, but we have mechanistically linked them to how they regulate response to clinically used antibiotics. And this was true for every single molecule that was only produced in the presence of antibiotic. Interestingly, multiple antibiotics with different mechanism of action, whether they target ribosome, whether they target membrane, whatnot, had the same effect. So many of these molecules are known antifungal compounds, and when patients have burkholderia in their lungs, they also have fungus in their lungs. So we thought maybe this is one way bacteria is interacting with the fungi. I'm not going to do a lot of biology here, just very quickly. Wild type does not kill the fungus. The fungus came from lungs of CF patients. The knockout does, because you've removed the repression. And if you add antibiotic, even the wild type starts killing, right? So we are adding antibiotics again to treat these patients, but they result in modification of bacterial fungal interactions within a human lung, okay? So with this, now let me summarize what I showed you after the first summary. So now I've showed you how untargeted metabolomics allows us to link mechanistic biochemistry with phenotype. So this really completes the circle, right? We have specific phenotypes of bacteria. We can link them to chemotypes using untargeted metabolomics. And then by analyzing the genomes, we can link chemotypes to genotypes. And then by using molecular biology, we can figure out how those are regulated, right? So by making gene knockouts, for example. And then determine their role in the function of bacteria in an infection-relevant model, okay? So with this, I would like to highlight a message that untargeted comparative metabolomics is a valuable approach as a hypothesis generator in model systems such as synthetic sputum that I showed, where you can detect microbial metabolites in high abundance, but you can mimic the infection-relevant environment to induce them to produce molecules they would be producing in a lung. And then you can test their relevance in ex vivo models, human samples, animal models. How much time do I have? I have plenty of time. So let me highlight how we test some of these hypotheses in ex vivo models. So our collaborator at Sandia National Lab cultured mammalian macrophages. So macrophages respond to infection. If you're an immunologist, you probably have studied macrophages for a while. So he cultured macrophages either by themselves or infected them with bacteria, such that bacteria is only living intracellularly in macrophages. So you get rid of any bacteria that's outside. So we are studying bacteria that's living inside human cells or mice cells. Now more than 50% of the metabolome was dysregulated upon introduction of bacteria. Same problem, we can't annotate anything, so what do? We combined a method called class enrichment with class prediction. So class enrichment was first described by Oliver Fien, but it was described for analysis where you've annotated molecules. So you've annotated molecules, you have their structures, and from the structures you predict chemical class, right? And then you do class enrichment. What chemical classes or biochemical classes are enriched in disease versus healthy? In our case, we can't annotate anything, or not very much. So we don't have structures to do class enrichment. So we thought we could combine cannabis predicted chemical classes to class enrichment. So we predict chemical classes using cannabis. So that uses MS2 spectra to predict chemical classes, and Jessica showed us that accuracy is 70 to 90% for chemical class prediction. So without annotating, you can get biochemical insights into your data. So shown here is enrichment. So red is enriched, blue is depleted. So when we compared our monocultures with pathogen co-cultures, these are the top chemical classes that were differentiating the healthy versus the diseased. So as an example, the top is prostaglandins. Prostaglandins are lipids that are produced at the site of infection. So right now, I don't have any prostaglandins on my skin. But if there was a wound which was infected, you'll see a huge signal from prostaglandins. So that's a good proof of principle that prostaglandins, upon bacterial introduction, were detected. So using such approaches, I don't have time, I didn't plan for it, but it looks like I have time, but I don't have a slide. We've found new molecules. For example, acylcarnitines are energy stores. They are important for function of mitochondria, for example. We found a completely new kind of acylcarnitines that are produced in response to infection. And that too in corals, right? So such kind of chemical class prediction allows you to take your annotations to the next step and discover new chemistry. Then we have these macrophage cell lines that have been infected with bacteria. We wanted to figure out what molecules bacteria is making. So prostaglandins are made by host, but we are interested in microbial metabolome, right? So when we looked at our data, we looked for peaks that were only in infected sample, right? Some of them are going to be prostaglandin host molecules, but others will be microbial molecules. So we found that some of the very low abundance peak in data-dependent acquisition, you don't even get MS2 spectra for them, right? So it's really difficult to annotate. And for any metabolite feature to pass our quality control, we require at least 2 MS2 spectra to be acquired. So we have some decent quality consensus MS2 spectra that we can search in the libraries, right? But then we combined this approach with with the previous model that I described, where we culture bacteria in a monoculture, we add antibiotics, we mimic the infection-relevant environment, right? So we have data from our monoculture. So when we have data in our monoculture, we see a lot of that molecule. We always get MS2 spectra. So we combine that data with this data. So now we have the UV visible spectra, we have MS1 spectra, we have retention time, and we have MS2 spectra. So this allows us to annotate these molecules as microbial molecules. So using this strategy, we annotated several microbial molecules, which are microbe-specific. These are not made by humans or mammals, to be produced intracellularly inside a macrophage cell line. To highlight how this is important, some of these molecules, for example, the ones highlighted in yellow here, were only produced in monoculture in response to antibiotics. In our macrophage cell line, we're not adding antibiotics. So that means mammalian cells are creating an environment that induces the production of these molecules. And the ones shown down here were called DACs. These are HDAC inhibitors, so histone deacetylase inhibitors. Histone HDAC inhibitors have been shown to repress the expression of phagocytic receptors on a macrophage cell line. So phagocytic receptors engulf the bacteria and help macrophages kill it. So if you repress the expression of phagocytic receptors, you can't engulf the bacteria, you can't kill it. So a few cells, bacterial cells, that are living inside macrophages are induced by macrophages to produce these compounds that prevents macrophages to kill them, right? So this allows us to now take our next step from microbe-microbe interactions to microbe-host interactions and figure out what pathways are important. And the genes for production of these compounds could be new drug targets to facilitate killing of bacteria in addition to antibiotics. With that, I plan to end my talk here. So a lot of work. Our undergrads really support us in looking for those spectra in databases. They sit hours and hours trying to find spectral matches in databases manually or in an automated fashion. Also, our collaborators who provide us with biology, biochemistry support as needed, funding. Thank you for your attention, and I'm happy to take any questions.
Okay, this time you don't have to come to the podium. I'll come to you.
Thanks so much for that talk. I thought it was very elegant. I know in the clinical space that typically trimethoprim is co-administered with sulfamethoxazole to increase efficacy. I was wondering if you could speak a little bit about the decision to either look at trimethoprim individually or whether you looked at the combination
We
drug.
should definitely look at co-administration. Initially, we started with trimethoprim for 2 reasons. Right, so when you administer both, you don't know you have a complex interaction, so you can't link to changes, and it's hard to predict mechanistic biochemistry. The second reason we chose trimethoprim just by itself was the work done by Mo Sayed-Sayemdost at Princeton University, where he developed this high throughput screening method where he would take these natural product compound libraries and he would culture bacteria in them. And he had tagged his bacteria with a GFP, and then he would look for expression of GFP. So what molecules change gene expression of this bacteria? And he found that trimethoprim induced the expression of, for example, GFP. So trimethoprim was found to be the robust inducer of these natural product virulence factor-like molecules. So that's one reason, but I agree with you. for a true clinical implication, we should add sulfamethoxazole in addition. It's just so we work at 10-fold lower MIC. So trimethoprim, let's say if it kills at 100 micromolar, we'll only add 10 micromolar to our culture because we don't want the bacteria to die. And that's clinically relevant because myself and other mass spectrometrists using imaging mass spec or other LCMS-based methods have shown that antibiotics do not penetrate uniformly into the lungs because of this thick mucus. So bacteria are exposed to sublethal concentration of antibiotics that allows them to develop resistance and then spread. So those were the several factors, but I agree 100% that we should repeat all of this with sulfamethoxazole in there.
Over here.
Thank you for that excellent talk. So you talked about untargeted metabolomics and ranking metabolites based on some aspects, picking these metabolites and ranking. So what are the different criteria that you would consider? Because often sometimes you'll be missing some important metabolites. So what is the basic criteria or criteria that you would consider to rank them?
So these are models like random. So the input to these models is your data, feature abundances, and then you classify your data into group A and group B. So group A is in presence of antibiotic, group B is in absence of antibiotic. And then these models will predict what molecules are different between group A and B. So they'll maximize the separation in the metabolomes of A and B and figure out what metabolites account for this separation. You may have seen loading plots in PCA. So if you see a PCA, you see these arrows going and what metabolites are describing those groups. PCA is not a good method to do that. Random forest, SVMs, PLS, regression. So those are the methods that are used for ranking the metabolites that differentiate your group A and B or explain the variance in group A and B.
I have one more question. Like you talked about co-culturing bacterial strain with the macrophage cell line.
Yes.
And you are interested only in looking for the intracellular molecules. But ideally, when we talk about quorum sensing molecules and all these bacteria, when they infect the host, they would secrete it outside the cell. So what made you...
Outside the bacterial cell, but they may still be inside the host cell.
Okay, so what was the rationale in selecting only the intracellular and not looking for the secretome?
Yeah, I didn't explain that. So Burkholderia are notorious and known to stay intracellular. So, extracellular are immune cells, antibiotics can kill them. But if they are intracellular, it's difficult to kill them. Most of the clinically used antibiotics do not penetrate macrophages or epithelial cell lines, so they don't enter our cells. They can enter bacterial cells. But Burkholderia overcomes that by its unique ability to grow inside macrophages. So for example, Bacillus cannot grow inside macrophage. Burkholderia is notorious for that. That's why we looked for intracellular metabolome.
Thank you.
So it's very interesting talk, and then I have a couple of questions. So first question is actually it's about quorum sensing.
Okay.
So first, contact of bacteria can be happen at mucin layers. So mucin contains a lot of mucin glycoproteins such as MAC5B, MAC5AC. So do you think those molecules can suppress quorum sensing gene or those production of metabolite?
So the mucin, you
Yeah.
mean? I didn't have time to show today. We actually, so when, we did this experiment in two different kinds of media. One was LBE, which is commonly used culture media for bacteria in the lab. The other was synthetic sputum media that contains mucin. We found a couple of interesting things that, the induction by antibiotics was much higher in actually synthetic sputum than in LBE. There could be a couple of reasons for this. Pseudomonas and Burkholderia are known to degrade mucin proteins, take the sugars off, eat them.
Yeah.
Secondly, synthetic sputum has much more methionine or amino acids in it, which we show that are important for the phenotype we see. So synthetic sputum does show, I guess because of mucins, a very unique phenotype because these bacteria can degrade mucin.
Okay, thank you. And then another question is, I may misunderstood about your talk today, and then I believe you mentioned this bacteria produces sulfur-containing metabolite.
Correct.
So do you think our body utilize those molecules to make more sulfated protein or sulfated glycans?
Probably. Maybe Josh can do some
So
proteomics.
because
Sulfa proteomics.
I also work with University of North Carolina Chapel,
Yes.
and then there is Cystic Fibrosis Center, right?
Yes.
I work with Rick Boucher. He is the director of that center. So recently profiled mucin glycans from the sputum.
Okay.
We see alteration of sulfation on the mucin glycans.
Oh, wow.
So I was wondering why those we see the difference of
Yeah.
sulfation on those molecules, and then
Could be methane thiol. So, people have done GCMS on breath of patients with cystic fibrosis, and they find a lot of methane thiol.
okay. And
Methane
then,
thiol is a biomarker. Bacterial produced methane thiol is a biomarker of CF.
Okay, great. And the last question is about macrophage data. You see increase of ceramide, right?
We do, always.
And then do you know what, have you looked at, ceramide synthase? Which genes, ceramide synthase 1 or 2 or 3, or what kind of group, ceramide has,
Yeah, that's
different.
a wonderful question. I
because
need a
Georgia Tech,
biologist.
that's one big name, Alfred Mail. I don't know if he's still working there, but, he used to study a lot of, study on the ceramide synthase, so.
Okay. Yeah, I need a biologist to answer that question. I don't know. But during my postdoc, we did metabolomics on sputum, longitudinal metabolomics on sputum. So exacerbation is when their disease is exacerbated, exaggerated, versus stable. And ceramides were the most important biomarkers of exacerbation. So
Thank you
maybe,
so
I
much,
don't
yeah.
know.
Okay.
Hi, great talk. I was just curious, you hinted at it earlier, what was unique about the acylcarnitines that you discovered in that co-culture experiment?
So acylcarnitines that have fatty acids with the acyl tail with the hydroxyl group. On the hydroxyl group, we found another fatty acid added. So they are called HFAs, fatty acid, hydroxyl fatty acid or something. We know fatty acids. So hydroxyl fatty acids would get esterification with another fatty acid on the hydroxyl group. And they have been biomarker of every single disease that have been studied. So we see those fatty acid esters of fatty acids added onto acylcarnitines.
This is more of a processing question. So you mentioned cannabis is your go-to for formula mass. Do you do that with every feature, or do you really focus on the things that you think are going to be different between your model system?
Depends on the question. So if the question is what chemical classes are enriched, then we do it on every single feature. But cannabis does not predict chemical classes, Jessica, correct me if I'm saying it wrong, greater than 800 MZ. And sometimes it's not confident in chemical formula prediction, then it won't give you chemical class. So it's not able to predict chemical class of every single feature, especially above 800 MZ, because you can't, even with the Orbitrap, for small molecules, it just gets too complex to predict chemical formulas. So depends on the question. If the question is just annotation, we want to annotate something, we don't annotate everything. What differentiates our condition A versus B is what we annotate. But if we are doing chemical class enrichment analysis, what chemical classes are enriched, then we do it on an entire data set. It can take up to a couple of days for it to run through the entire data set.